Hi guys, we're here for the last part of our photosynthesis notes, so let me get that shared. And that was our notes from last time, so we're done with those. And now we're going to our last set of photosynthesis notes. And here we're going to look at the Calvin cycle. This is also often referred to as light-independent reactions or dark reactions. These do not require light. They're going to use the materials that we got from the light reaction. So light came in, water came in, we got oxygen out, and we made ATP and NADPH. So now we're going to take the energy from ATP and NADPH, combine it with carbon dioxide to create sugar. The Calvin cycle is a cyclical electron flow. So we're going to start with a certain molecule. We're going to get the electrons and we're going to process through the electrons and it's going to come back to that original molecule. We use ATP and NADPH to reduce carbon dioxide. Yes, that is a typo. I'll need to fix that, but it should be NADPH. You'll see it in the actual diagram. To reduce carbon dioxide to sugar. For a net synthesis of one G3P or sugar molecule, the cycle must take place 3 times. So we have to go through the Calvin cycle 3 times to get one single sugar molecule. And here you can see we've got a lot of specific molecules. You don't need to know the individual molecules and individual detailed steps within the Calvin cycle. We're just kind of looking at the big picture, remember? So we have 3 phases in the Calvin cycle. We have what we call carbon fixation. We have reduction, and we have regeneration of RUBP. Obviously, we need to recreate a molecule that we started with. And so throughout the process, otherwise it wouldn't be a cycle if we didn't recreate it, right? Okay, so phase 1 is carbon fixation. So the carbon dioxide becomes incorporated into the Calvin cycle one at a time. We're going to bring one carbon dioxide in at a time to use. Each carbon dioxide is going to be attached to a molecule of RUBP. That's the initial substance in the carbon cycle that we're going to work from. It is catalyzed by an enzyme known as Rubisco. So remember, enzymes are what makes it possible for life to happen because it makes the reactions happen faster. This is going to form something called 3-phosphoglycerate. Phase 2 is our reduction. So we're going to take that 3-phosphoglycerate and it's going to be phosphorylated by ATP. We're going to actually add phosphate to an energy to the phosphoglycerate. And it said, notice it says it uses 6 total. So we got to have 6 of these ATPs to make this process happen. It then becomes what we call 1,3-biphosphoglycerate. 6 NADPH molecules are then going to donate electrons to this 1,3-biphosphoglycerate, which reduces it to G3P. 6 molecules of G3P are going to be formed, but only one is actually counted as a net gain. The other ones are actually going to be utilized to reform our RUBP. So the other 5 are going to regenerate the RUBP so we can go through this process again. So you see why we have to keep going around and around just to make one glucose. So phase 3 is the regeneration of RUBP. And those 5 G3P molecules that were left are going to be used to regenerate 3 molecules of RUBP. We use 3 ATPs for this regeneration. So it's going to take some more of our energy that we made in the light reactions to reset this to run the system again. Once we have RUBP recreated, we're ready to take in the carbon dioxide again and go through the process. So around and around. So here's our RUBP. We're going to add the carbon dioxide. We're going to then put ATP to add more phosphate to it. We're going to add the electrons from NADPH. One of our G3P molecules is going to get kicked out. The other ones are going to continue around, and we use ATP to regenerate our RUBP. So in terms of what do we get, what do we put in? We put in 3 carbon dioxides, 9 ATPs, and 6 NADPHs. And remember, ATP and NADPH came from our light-dependent reactions. And the plant took in like it inhaled, but not really. Remember, through the stomata, it allows it to pass, took in carbon dioxide. The outputs, we get 1 G3P, so 1 sugar, 9 ADP, and 6 NADP. These, the ADP and NADP, will go back to the light-dependent reaction to be recycled. So we use NADPH, ATP, and carbon dioxide. We produce a 3-carbon sugar called G3P. Well, glucose has 6 carbons, so that's why we have to do it twice. Remember, we said that. There are 3 phases: carbon fixation, that's where we bring the carbon dioxide in. Reduction, that's where we take the energy from the ATP and from the NADPH to make our G3P. And then we take the remaining 5 G3Ps and use them to regenerate RUBP, also using ATP. So the reality is not all plants work exactly the same, because not all plants live in the exact same environment and situations. So we need to account for some of these variations. We have a type of plants called C3 plants, and they're going to use photorespiration. So on very hot days, they actually close their stomata so that they're not losing water. Because remember, I mentioned that water also passes through the stomata through a process called transpiration. That's something we looked at when we did the water cycle. So when it's really, really hot, we're going to have a lot of water evaporating this way, and we don't want that. So they're going to close the stomata. The problem is, if you close the stomata, are you exchanging carbon dioxide and oxygen, those other gases? So that means that we're going to have less carbon dioxide present and more oxygen present, because we can't exchange those if the stomata is closed. So Rubisco will actually bind to the O2 and use ATP to produce carbon dioxide. So we can take a piece, a segment in the carbon cycle, and using a little extra ATP, we can produce the carbon dioxide that we need. The problem is that no sugar will be produced in this process. It's extremely bad for the plant because it needs sugar to live, just like we do. So in terms of adaptations, plants that live in hot, dry climates have actually evolved alternative mechanisms of carbon fixation, so we don't have that problem. C4 plants have spatial separation of steps. So the steps are actually taking place kind of further away from each other. They also, the stomata partially closes to conserve water, but it's not completely closed. And mesophyll cells can actually fix CO2 into a 4-carbon molecule, so we can kind of have that carbon preset for the cycle. So it's then transferred to a bundle sheath cell, which releases the carbon dioxide to be used in the Calvin cycle. So these cells can actually take a whole lot and basically pre-create 4-carbon molecules, which then it can use for the carbon cycle. Maize, corn, a lot of grasses, sugarcane would be one that's really common to use this C4 plant adaptation situation to try and deal with the carbon dioxide closing the stomata extremely hot atmosphere concept. And then we have what we call the CAM plants. These will open the stomata at night and close it during the day. Obviously, at night when it's cooler, there's less of an issue of transpiration, loss of water. Carbon dioxide is incorporated into organic acids and stored in the vacuole. So it actually stores the carbon dioxide in the materials in the vacuole during the night when it has the stomata open. And then during the day, it's able to use the carbon dioxide that was stored in the vacuole for the Calvin cycle. Pineapples, cacti, succulents, jade, these are just some examples of what we consider CAM plants. Okay, we have a practice-free response question. A student wanted to study the effect of varying colors of light on the growth of plants. The student exposed tomato plants to 3 different wavelengths of light for the course of 1 week and measured the overall height each plant each day. All other conditions were kept consistent between the plants, and the results are below. And so here you'll see our graph. We have the 3 different wavelengths, and we have the height by the number of days. Identify the wavelength that caused the least amount of growth. Explain why the wavelength identified in part was ineffective. Okay. So hopefully, here's our next one. I have to move me. Rubisco can bind to CO2 or O2 in the Calvin cycle. After learning this, a student was interested in photorespiration. After doing research, she claimed that it is simply a metabolic relic. Early atmospheric conditions did not have oxygen. Therefore, it never posed a problem early on. Would you support or reject this claim? Justify your response. So you're going to have to kind of go back to the beginning of our photosynthesis notes when we talked about the cyanobacteria to guide you here a little bit. Okay, so hopefully that has helped you understand the concept of photosynthesis. And we'll do some other activities and work with it. Bye.